welcome to this, our latest Saints Talk live from St Andrews. My name is Alex Hayes. I'm Head of Development for Arts and Divinity at the University, and I'm delighted to be introducing today's talk by Dr Julie Oswald, which is Can You Hear Me Now? Using Sound to Study Marine Mammals Around the World. Dr Oswald is a Senior Lecturer in the School of Biology at the University of St Andrews and Deputy Director of the Scottish Oceans Institute, where she is a member of the world's largest sea mammal research unit. She is also director of the university's vertically integrated projects program, which takes an innovative and inclusive approach to research projects across the institution. Her own research focuses on acoustic signals that communicate information such as species identity in dolphins and the development of acoustic species classifiers. She was recently awarded the Allen Institute Conservation Technology Award for her work using AI to identify dolphin species in acoustic recordings, and you'll hear a bit more about that very shortly. Marine mammals are important species in marine ecosystems, and many face serious threats to their survival. Studying the distribution, abundance, and behavior of these animals can be challenging because they spend so much of their time underwater and many live in areas that are difficult to access. Fortunately, however, many marine mammals produce sounds, and over the past several decades, marine scientists have been using passive acoustic methods to investigate many aspects of the lives of various species. Dr. Oswald will discuss the latest ways in which she uses sound to study marine mammals, how the challenges of identifying species can be overcome by using machine learning methods, and what she and her colleagues have learned from applying these methods to passive acoustic recordings of marine mammals. Now, I'm excited to share with you that the talk you're about to hear may include instances of marine mammalian creaking, clicking, and possibly whistling, some of which could be oscillatory, and I'm finding that all rather exciting. Julie will speak for around 30 minutes, after which you'll have the opportunity to ask her questions, and I would vigorously encourage you to take that opportunity. Please do submit your questions in the chat, and you can do that at any point during the talk. Having hosted a few of these, I have noticed a pattern where questions can be a little slow to emerge at the start, and then we get a deluge with 2 minutes to go, which means we're not able to include them all. So please don't hold back in submitting your questions and just put them in the chat as they occur to you. And we will wrap up promptly no later than 6:15 BST, so in just under 1 hour's time. So without further ado, I'll now hand over to Dr. Julie Oswald for her talk, Can You Hear Me Now? Using Sound to Study Marine Mammals Around the World. Julie.
Thanks so much, Alex, and thank you to everybody who's joined to watch this talk. Today, I'd like to talk to you a little bit about some of my work that focuses on using acoustics to study marine mammals. Now, with the growing concerns about human impacts on our planet, and particularly our oceans, ecology and conservation biology have become a key focus of efforts to protect animal species and their habitats. Now, almost the very first questions that are asked when trying to manage an ecosystem are, what species are present, in what numbers are they there, and how do they use their environment? Now, biologists have primarily relied on visual surveys to gather this information. These surveys can take place from ships, from airplanes, from under the water, and even from land. And they can provide us with a lot of important information, like what species are present, how many individuals are there, what are they doing, and how healthy are they. So this is a really great tool for us to use. However, visual surveys are constrained. They're constrained by darkness, by high sea state, by rain, by fog, and even by animal behavior. Luckily, many marine species produce sounds, and so we can use acoustic methods known as passive acoustic monitoring to listen to them and survey these species. Now, there are a lot of different methods that we can use to collect passive acoustic data. For example, we can make recordings from boats using underwater microphones called hydrophones. We can put recording packages on the seafloor and leave them there to record for days, weeks, or even months, and then come back and retrieve the data later. There are seafloor mounted hydrophones that send data back in near real time via satellite links or underwater cables. And we can even put tags on the animals and record them that way. So there's a lot of different ways to collect this acoustic data from marine organisms. And PAM methods provide some really significant advantages. They're not constrained by weather or darkness, and they give us the capability to monitor for a long-term, continuous amounts of time, and especially in remote locations that might be difficult to access otherwise. However, like everything else, passive acoustic monitoring also has its limitations. And one of the biggest limitations is, of course, that not every species produces sounds. And of course, we can only record species that are producing sounds. So because of this, many PAM studies focus on indicator species. Now, indicator species are those that are among the first to be affected by changes in the environment. And so monitoring their presence, absence, and abundance can provide critical information about ecosystem health. Cetaceans, which are the whales and dolphins, are important indicator species, and they produce a lot of sound, so they're excellent subjects for passive acoustic monitoring. In my work, I focus on dolphins. Dolphin species produce a lot of different kinds of sounds. And these sounds are grouped into three main sound types. They use whistle sounds. These are illustrated here on this graph. This graph is a spectrogram. A spectrogram is a visual representation of sound. So on this graph, we have time on the x-axis and frequency, or the pitch of the sound, on the y-axis. And then the loudness of the sound is represented by color. So in this spectrogram, the loudest sounds are the red colors. So the whistles are represented by these squiggly lines on the spectrogram. And whistles are used by dolphins for communication. Another sound type that dolphins produce are echolocation clicks. And you can see these on the spectrogram as these thin vertical lines. Echolocation clicks are the dolphin's sonar. So they're using these clicks to see their environment using sound. And finally, dolphins produce burst pulse sounds. Burst pulses are clicks that are really, really close together. And they're so close together, in fact, that our human ears can't discern the individual clicks, and they sound and look on the spectrogram like tonal signals. So these sounds all sound quite distinct from one another and can be really fun to listen to. So as Alex promised, here is some sounds produced by bottlenose dolphins. This is the sounds in the spectrogram you're looking at, and they were recorded up in the Moray Firth. So hopefully this will work. So dolphins can be really noisy, and it's great to study them acoustically, but a lot of the acoustic recordings that we collect don't have visual observations associated with them. Or even if they do have visual observations, we can't always see the animals that are producing the sound. So it's very important for us to be able to look or listen to these sounds and identify what species is producing them. But this is where one of the big challenges lie. It can be really difficult to identify dolphin sounds to species. And this is for two main reasons. The first is that dolphins produce so many different kinds of sounds. So here are four more spectrograms. They're four spectrograms of whistles. The whistles are the squiggly white lines. And these whistles all look quite different from one another. And they were all recorded from one school of striped dolphins. And this is just a really small sample of all of the different whistles that were produced by this one school of dolphins. So every species of dolphin that whistle produces a lot of different kinds of whistles. So this huge variability can make it difficult to pinpoint what is characteristic of that species. And then to make things even more difficult, there's often a lot of overlap in the characteristics of the whistles produced by different species. So this top spectrogram is a whistle produced by a spinner dolphin, and the bottom is a whistle produced by a rough-toothed dolphin. These two whistles have very similar shapes, they have similar frequency ranges and similar durations, so it can be really difficult to say which species produced them just by listening to them or by looking at the spectrogram. So this brings us to the need for acoustic species classifiers. These are statistical tools that we use to identify species in acoustic recordings. These can be statistical classifiers using things like linear discriminant function analysis, support vector machines, or random forest analysis. And these analyses all take in measurements from the whistles and use them to uncover the subtle differences between species. So as input to these classifiers, we often use variables measured from spectrograms. So if we look at this spectrogram of a whistle, you might be able to see that there's a lot of different things that we could measure from the spectrogram. For instance, we can measure the frequency at the beginning or the ending of the whistle. We could measure the lowest and the highest frequency in the whistle. You can measure how long the whistle is. You can measure slope of the whistle, how often the slope changes. Lots of different variables that you can measure to describe the shape of the whistle contour. Now, more recently, there's been a huge explosion in the use of machine learning and AI methods to create classifiers. These methods are things like artificial neural networks and deep learning techniques. And these methods use a variety of different inputs. Sometimes they use spectrographic measurements, just like the statistical classifiers, but they can also use things like images of spectrograms or even raw audio files. Now, in my work, I've focused mainly on random forest analysis and more recently, some deep learning neural networks. So I want to just spend a few minutes explaining to you how these two analyses work before we go any further. So a random forest is a collection of decision trees, like the one illustrated here, where the data are divided into the most parsimonious groups through a series of binary splits. So each one of these splits is based on the value of one of the variables that you've measured from the spectrogram. So for example, if you look at the tree here, a whistle will start at the top of the tree, and if it has a minimum frequency of less than or equal to 11 kilohertz, it'll go down to your left. Or if it has a minimum frequency of greater than 11 kilohertz, it'll go down to the right. And it just continues on down the tree until it reaches a terminal node, which is one of these orange boxes, where a classification is reached. Now, a random forest is composed of hundreds or even thousands of these classification trees. And each tree is different from all of the other trees. And this is because at each one of these nodes, the blue circles where the data are split, the variable used to split the data is chosen from a random subset of all of the variables that you've measured. So this injects some randomness into the tree growing process and means that all of your trees are different from all of your other trees. So now when you have a new whistle to be classified, it's fed through each one of the trees in the forest. Each tree predicts a species, and then the whistle is classified as the species that got the greatest number of tree votes. So this can be a really powerful tool for species identification. And then a bit newer technique is deep learning. Now, deep learning using neural networks enables mapping of complex relationships between inputs to the model, so in our case, the whistle variables, and the outputs, in our case, the species. Now, the inputs are associated with the outputs through a series of weights and functions. So this illustration here is a simple neural network. And in this neural network, we have our inputs, our whistle variables, and multiple weight functions are calculated for each input, and these are translated into activation functions. Now, the results of these activation functions are called hidden units. And they're called hidden because there's no actual observational values for these values in our data. So we haven't actually measured them. They're just calculations that have been made based on all of these weights. These hidden units are then used as input to the second layer of your neural network. And the second layer is the layer that predicts the class or the species. So training a neural network like this involves modifying the weight functions iteratively to maximize the output accuracy or the ability to predict species. So essentially, the computer is learning what the best functions are. And more complicated neural networks involve more layers and different functions. More layers can provide greater accuracy, but require a large amount of data. So no matter what kind of classifier you're going to use, the first step is to collect a very large data set for training the classifier. We need very large data sets regardless of the method that we're using because of all the variability in the vocalizations that these animals produce. You want to capture as much of the variability as possible in order to identify the species accurately. You need a very large data set to train your classifier, but you also need a data set to test your classifier to see how well it does on new independent data. Now, your data also need to have visual confirmation of species identity. Any sound that you put into your training data set, you have to know what species produces that sound. It's important to have visual observations with this so that you know that the sounds that you're putting into your classifier are being trained accurately. These sounds also need to be recorded from single species schools with no other whistling species nearby at the same time. This is because even if you're watching the dolphins, they often produce sounds when they're underwater and you can't see them. But if you can see them, they don't move their mouths when they produce sounds. So it's really hard to tell which animal is producing the sound. So if you have more than one species in the area, you don't know which species is producing the sound, and you can't use that sound to train your classifier. So it can take a really long time to collect these large data sets that you need to train and test these classifiers. Another thing that's common to all classification approaches is the need to develop classifiers that are specific to the area in which they will be used. Now, this is because different locations contain different collections of species, and including species that are not present can introduce error into your classifier. Also, individual species that are found in different locations often sound different in those different locations. So this is akin to having accents, although we scientists like to call it geographic variation. Now, one of my PhD students, Charlie Hargrave, is currently exploring geographic variation in the whistles produced by bottlenose dolphins. And he's found that even bottlenose dolphins on the east coast and the west coast of Scotland sound different from one another. Now, I've been working on classifiers for quite some time, and I'm working towards building location-specific classifiers for the entire world. When I started doing this as a PhD student at Scripps Institution of Oceanography, I created a classifier for the Eastern Tropical Pacific Ocean. I then continued my classifier work as a postdoc at the University of Hawaii, and then later running a bioacoustics consulting company in San Diego. And during this time, I expanded my library of classifiers to include both the East and West coasts of the United States and a more targeted classifier focused on species around the Hawaiian Islands. And now that I'm in the UK, I'm working on classifiers for this side of the world. One of my PhD students, Tristan Klein, has just completed the development of classifiers for the Northeast Atlantic and the North Sea. And I'm currently also working on classifiers for the Mediterranean, Macaronesia, the waters off of West Africa, and the Southeast Atlantic. So I'm getting close to my goal of world classifier domination, but I've still got a ways to go. Now, my classifiers are all available in a module called RACA, which stands for the Real-Time Odontocetes Call Classification Algorithm. And this sits in a free open source acoustic software platform called PAMGuard. Both PAMGuard and RACA are being used by researchers, government agencies, and companies worldwide for a variety of applications. Now, I don't have time to talk about all of the classifiers or all of their applications right now, but I do want to talk to you about 3 case studies from my more recent work to show you how all of these classifiers can be applied. So first, I'll talk to you about a case study having to do with monitoring populations off the east coast of Scotland. And then we'll talk about a conservation example focused on the west coast of Africa. And finally, I want to talk to you about an education example, how I'm applying my classifiers in the classroom. So first, the case study. The east coast of Scotland is home to 7 delphinid species. So these include bottlenose, common, and Risso's dolphins, white-beaked and white-sided dolphins, and pilot whales and killer whales. Now, these guys are called whales, but they are, in fact, dolphins. So the east coast of Scotland is also the site of a lot of anthropogenic activity. We've got things like fishing, shipping, offshore oil and gas production, and offshore wind installations. And all of these things can have an impact on these dolphin species. So because of this, we have a great need to monitor these populations. We need to know where they are, when they're there, and how they react to the things that we're doing in the ocean. So to that end, Marine Scotland has been running a project called the East Coast Marine Mammal Acoustic Study, or ECOMAS. And as part of that study, they've deployed 10 seafloor-mounted acoustic recorders along the east coast of Scotland. And the locations of those recorders are pictured in this map here. Each one of the red dots is a location of one of the recorders, and they range from St. Abbs in the south all the way north to Latheron. These recorders are in the water and recording every year between March and November. And this has been taking place since 2013. So this is a really large and rich data set that we can use to monitor cetacean presence as well as anthropogenic noise. And one of the big motivations for this was to look at whether dolphins are affected by the construction of offshore wind. So we're looking at the presence of dolphins before, during, and after these offshore wind installations go in. But until now, it's only been possible to look at dolphin presence as a whole, and we couldn't look at species-specific patterns. But now we have Tristan's new classifier for the Northeast Atlantic and the North Sea. And Tristan has spent some time applying this classifier to recordings that were made in 2019. Tristan's classifier is a deep learning classifier, like the deep learning methods I described to you earlier. It includes all 7 species that are found in the Northeast Atlantic, and it consists of deep convoluted neural networks trained using 70,000 whistles and clicks from 105 different encounters of dolphins. And all of these encounters had visual confirmation of species identity. So this was a huge data set. The way the CNN works is that it takes brief clips of sound and classifies those to species. And then the output, all of these classifications are then fed into a second step, a random forest classifier, to classify entire encounters or schools of dolphins based on individual classifications produced by that school. And Tristan's classifier has a really high accuracy. 86% of encounters or schools are correctly classified. Now, this is really a great classification result, especially when you consider that when you have 7 species in your analysis, you'd expect 14% of those to be correctly classified just by chance alone. So being able to classify 86% of encounters correctly is a great improvement over that. Now, another way to look at the success of a classifier is to look at this type of table, which is called a confusion matrix. I know there's a lot of numbers here. I'm going to walk you through it. Don't worry. So the blue numbers here are the correct classification scores for each species. So the percentage of encounters that were correctly classified for each one of the species in the analysis. And what we see here is that correct classifications were high for every species. They ranged from 80% for common dolphins all the way up to 92% for white-beaked dolphins. So this is a great result. looking at a confusion matrix can also help us to understand where the errors are made. And this can help us then to interpret the results that we get back when we apply the classifier to new data. So for example, if we look at the misclassifications, that's all of the white boxes, we can see that killer whales and pilot whales are most often misclassified as one another. And Atlantic white-sided dolphins and common dolphins are most often misclassified as one another. So we can use this information to help us to interpret the results of applying the classifier. So when we did apply the classifier, this is the kind of information we get out. It allows us to look at both spatial and temporal patterns in the distribution of species along the East Coast. So these graphs are showing us the number of days per week that had events classified as each species. So day of the year is along the x-axis, and days per week with events is on the y-axis. Each one of the graphs is a different location, a different one of our recorders, and each species is represented by a different color. The gray boxes are the times when the recorders were on. So sometimes they weren't on for some period of time because something failed or they had to be taken out of the water for some reason. But if we look at all of these graphs together, I know there's a lot of information here, so I'm just going to give you the highlights. But what we can see is that there's a lot of variability in the number of days with acoustic detections across the different sites. So we have very few detections at Arbroath, Helmsdale, and Spey Bay, and quite a lot of detections at Cruden Bay, Latheron, St. Abbs, and Stonehaven. So now you know the hot spots. Now you know where to go and find dolphins. So that's looking at things overall as a whole. But if we look now at a specific species, I want you to focus on the brown bars. The brown bars are bottlenose dolphins. And what we see when we look at bottlenose dolphins is that this is the most commonly identified species at most of the sites. And where bottlenose dolphins were detected, they were generally detected through the entire study period, so from March to November. Now, the only exception to this was Latheron. Remember, Latheron's our northernmost site, and at Latheron, it wasn't bottlenose dolphins that were most commonly detected, but it was Rizzo's dolphins. Rizzo's dolphins are represented by the orange bars here. Another difference we see is that Rizzo dolphins weren't detected throughout the entire study period. There were actually two peaks in detections, one in May and the second from September to November. And if we keep looking at this Latheron site, another interesting thing pops out. If you look at the green bars, those are white-beaked dolphins. And we can see that white-beaked dolphins were not detected very often anywhere, but when they were detected, it was most often at Latheron and most often between September and November. So that's a really interesting result for that species. Now, we could look at broad scale occurrence patterns, but we can also look at things more fine scale, looking at data like this. This is now looking at the hour of day when detections were occurring. So you have hour of day along the x-axis and the relative density of classifications. So this is different species were detected relative to one another. Again, we have the different species represented by different colors. And what we see when we look at this graph is that for most species, most of the detections occurred at night. So the period between 6 in the evening and 4 in the morning contained over half of the detections for common dolphins, bottlenose dolphins, Atlantic white-sided dolphins, and Rizzo's dolphins. And for all of these species, there were two peaks in detection, one between 7 p.m. and 10 p.m., and the other between 1 a.m. and 4 a.m. And then when we compare this to the green line is our white-beaked dolphins now. And for white-beaked dolphins, they follow a different pattern. Most of their detections occurred in the morning. In fact, 67% of white-beaked dolphin detections occurred between 4 a.m. and noon. And for the other species, less than a quarter of their detections occurred during this time period. So this analysis lets us understand some really important things about the presence of the different dolphin species in the different locations. Now, when we're looking at these results, it's important to keep a couple of things in mind. First, it's important to remember that a lack of detections doesn't necessarily mean a lack of animals. Dolphins do produce a lot of sound, but they're not all vocalizing all the time. So it is possible that animals are present, but just being quiet. And so we need to do a lot more research on how often different species vocalize. And we need to think about and learn about how this changes with things like species, behavior, location, and time of day. It's also important to remember that there will always be some misclassifications. No classifier is perfect, and so it's important to understand these patterns of misclassification that I showed you in the confusion matrix. And understanding these patterns enables us to interpret the results more accurately. But even with these caveats, this type of analysis provides us with a much more complete understanding of which species are occurring in which areas and when they're there. And the prevalence of detections of most species during the nighttime hours highlights the importance of monitoring around the clock and not relying solely on visual methods. And data like this is crucial for things like the development of conservation strategies like marine protected areas and for monitoring the impact of human activities such as offshore wind developments. Now, we're currently preparing scientific publications on the classifier and its application. But if you're excited about the classifier and you'd like to learn more, it has been published on Zenodo, so you can go to this website and check it out for yourself. Okay, moving on to case study number 2. This is a conservation case study based on the Atlantic humpback dolphin off the west coast of Africa. Now, the Atlantic humpback dolphin is a critically endangered species that lives in the coastal waters off the west coast of Africa. In this region, it faces many threats, including bycatch, hunting, and habitat degradation. But we know very little about humpback dolphins. For example, we don't know how many there are, and we don't know if it's one continuous population or several small pockets of populations. What we do know is some of the distribution of Atlantic humpback dolphins. So this map of Africa shows in blue the countries that have confirmed records of Atlantic humpback dolphins, and in gray are species that don't have confirmed records of Atlantic humpback dolphins. Most of these confirmed records come from bycatch records, hunting, or opportunistic observations, people just happening to see and report humpback dolphins. So there's very little research happening on the Atlantic humpback dolphin because of limited resources and capacity in these countries. And so it's very difficult to create conservation plans for a species that so little is known about. Now, recently, the Consortium for the Conservation of Humpback Dolphins began using acoustic recorders to gather more information about this species. This is great, but there are several other species in the area that produce similar sounds to the Atlantic humpback dolphin. These are bottlenose dolphins, common dolphins, and even manatees produce sounds that are similar to dolphins. So it can be difficult to take these recordings and say for certain what species is in them. And this is where I come in. So I'm currently collaborating with CCAD researchers in Senegal, Gabon, Guinea, and Cameroon to collect visually validated acoustic recordings of all of these species so that I can then use them to train a new classifier. These photographs show my PhD student Tristan Klein, who was in Senegal earlier this year to help collect some of these acoustic recordings, as well as to train the local researchers there in how to collect the recordings after he leaves. Now, once we have enough recordings and I'm able to create a classifier, this classifier will be incorporated into Raka, so it'll be freely available for all to use. And I find this such an exciting project because not only will it have significant conservation implications, but it also allows capacity building in the West African countries. I'm working with the local researchers to equip and train them to collect passive acoustic data and also to use classifiers and interpret the results they get. This is a long-term project because resources are limited for data collection, but I'm really excited about the progress we've made so far, and stay tuned for some exciting updates in the future. And this brings me to my last case study, education. So I have integrated my research into my teaching here at St. Andrews by developing a vertically integrated projects module that teaches undergraduates in biology, computer science, and maths and stats how to do this work. Now, VIPs are credit-bearing modules where students from different schools and different years all work together on real-world ongoing research. This teaches the students really impactful research skills. For example, my students are analyzing acoustic recordings, they're comparing different classification methods, and they're developing new software tools for data management, for classification, for data analysis. So they're really working to push this research forward. They're creating new knowledge and new tools, which is really exciting for both them and for me. But not only does this teach the students research skills, but they're also developing really important employability skills. They're developing things like teamwork, communicating across disciplines, time management, project development, all things that can be really difficult to develop in traditional lecture-based modules. These pictures are all of my students from this past semester from my VIP. My co-supervisors and I were able to take them to an undergraduate research conference in Newcastle, where they presented 2 posters and 2 talks on the research that they've been doing in this project. And this is an amazing learning experience, not only for them, but also for us. And I love this way of teaching so much that I've taken on the directorship of the VIP program. And we currently have 15 VIPs running across the school, across the university in many different schools. And we're on track to having 20 VIPs by semester 2 of next year. So I feel like I've really come full circle from being a PhD student creating classifiers to expanding my library and applying these classifiers to now teaching students in this work. And I find this to be one of the most rewarding aspects of engaging with science. So now I'd just like to thank you all for your attention, and I wonder if anybody has any questions.
Julie, thank you very much indeed. That's absolutely fascinating and slightly kind of mind-boggling, both in terms of the complexity of the data inputs and the scale of the data gathering operation. I'm delighted to say that we have a number of very good questions, so I'm going to dive straight in with these. The first one comes from Sebastian Roberts. And this was actually the question that was occurring to me as you were talking. Could changing geographic variation in specific areas over time cause classifiers to need updating at intervals to maintain accuracy? And if so, how often?
That is a fabulous question. We know that the calls of killer whales do change over time. We don't know as much about how the calls of other species change over time. This is a question that I've talked about a number of times with people, and it's something we really need to look into. We have some data from Rizzo's dolphins that were collected back in the 1980s, and some new data that I'm really hoping to explore and look at how these vocalizations are changing over time. And if they're changing significantly, then yes, it does mean that classifiers will need to be updated. And I guess that's a little bit of job security for me.
And is the reason that there's some understanding of killer whale variation? Is that because the longevity of the species and you've got individuals that have been and groups that have been studied over a long period of time, or what's going on there?
Partially that, and partially because there are populations, particularly off the west coast of North America, where these matrilineal pods produce a distinctive vocalization. So you can identify a pod based on the vocalizations that they're producing. Unlike many of my dolphin species, the killer whales produce these distinctive sounds. And so by monitoring these stereotype sounds that are produced by the pods, you can really see how they're changing over time. And there's quite a lot of research going on into those guys.
Fascinating. Okay, next question. This is from an anonymous questioner. So do these neural networks act as language detectors here? That is, can it identify, sorry, something. Can it identify for a fixed species a set of alphabet sounds?
Mm. I knew that question was going to come. Yeah, so language, as we know, language involves syntax, it involves words, and this is not something that we have found so far in all of the acoustic study that we've done up until now. Now, deep learning does give us the capacity to find more detail, finer scale differences and patterns in the data. So I'm sure that we're going to learn new things about the communication systems of these species. But the fact that we haven't found any evidence of language up until the present time suggests to me that language as we know it doesn't exist. That doesn't mean that different sounds don't have different meanings, and we already know some of the meanings behind some of the sounds. But language, the way humans use language, is probably not happening in these animals.
Okay, interesting. Thank you. Question from Abigail Motor. I hope I pronounced that right. How have policymakers integrated your and your PhD students' findings into their decision making?
Great question, Abigail. So my classifiers have been used by NOAA and in the United States on population abundance surveys. And
Can you
so
just explain what NOAA is for
Oh,
those of
sure.
us who?
It's the National Oceanographic and Atmospheric Administration, and NOAA is in charge of monitoring and taking care of populations. One of the many things that NOAA does is monitor marine mammal populations. And so my classifiers have been used during these population abundance surveys, and they help to understand how many schools the visual observers are missing. The population size estimates are all based on visual data, but we know that not all schools are sighted. So the acoustic classifiers can help us to understand how many schools are missed, and that gets all incorporated into these equations to calculate species abundance. The newer classifiers, Tristan's classifier for the Northeast Atlantic, this work that he's done to apply the classifier to the ecomass data, we hope is the first step in informing new policies and helping us to understand the distribution of dolphins. This was a very first step. Nobody's been able to analyze this data like this before, and I think we've really proven that it's useful and important. And so I hope that by analyzing more data, we'll be able to incorporate this information into policy.
Thank you. Next question from another anonymous questioner. These are open ocean data. Could they be compared to data gathered in some enclosure so that the species is already known and extraneous sounds are not prominent?
Yeah, so that's another great question. So if you mean species in enclosures, if you mean species in captivity, then yes, there's been some work looking at whistles produced by dolphins in captivity and in the wild, and there are some similarities and differences, but because of things like geographic variation, there's always going to be differences in the whistles produced by these species. And so a lot more work needs to be done to understand the scope of this variation and whether you can use recordings made in captivity to create classifiers for dolphins in the wild. Another limiting factor is the species. So there's quite a number of bottlenose dolphins that we can go and record in facilities, but there's not a lot of the other species that we can go and make recordings of.
Mm-hmm. Thank you. Question from Harry Zaimovich. Hi, Julie. Thank you for such an interesting talk. Are these recordings across Scotland running 24/7? And if so, how does one pass all of that data across 10 locations? Can it be run through a script, or does someone need to go through that manually?
That's another great question. Thanks, Harry. So the recorders are running 24/7. but they're running on a duty cycle. So a duty cycle means that they record for some period of time and then they're off for some period of time. So for example, they're recording for 10 minutes every 30 minutes. So you're not getting 24 hours of recording each day. It's still a lot of data. So the way that Tristan's classifier works is it works on automated detections. So PAMGuard, one of the great things that PAMGuard has is automated detectors for both whistles and clicks. So you can run the recordings through these automated detectors and get results a lot more quickly and with a lot less headache, usually, than you could doing it manually.
Got it. So that's sort of, so I guess it's a bit of both is what you're saying. Yeah.
Yeah, a little bit of both. Even when you run a detector, you need to ground truth that detector, see how well it's performing. So there's always some level of manual analysis that has to happen.
Right. Next one isn't a question. It's just a big thank you from the mum of one of your VIP students.
Oh,
so that's nice.
so nice.
Next question is this data refined enough? Again, this is from an anonymous question. Is this data refined enough to be able to determine whether communication is taking place between different animals nearby? Also, how close is nearby? Can sounds travel and how far?
Big questions. So communication between animals nearby. So the data that I use, we can't identify the individuals producing each sound. So it's very difficult to look at the animal that's producing the sound and the animal that's receiving the sound and understand if information has been transferred. For that kind of study, you need to be able to really see the animals, see their behavior, and localize the sound to individuals. So that's a really difficult thing to do. It's possible in some locations where waters are clear and animals hang around, but not with my data. What was the other question? Oh, how far can they travel? So yeah, that's another great question. How far whistles travel depends a lot on the propagation characteristics of the location you're in. As a general rule of thumb, based on all of the work that I've done and some propagation modeling I've done, I use 3 nautical miles. So another school cannot be cited within 3 nautical miles to be used in the classifier.
Interesting. I remember speaking to a colleague of yours not that long ago about whale communication, and that there was some thought that maybe whale hearing isn't quite as good as might previously have been thought, and that the fact people have, or researchers have assumed that just because whale sounds travel for enormous distance underwater, that means that they're being heard and performing a function, when that may in fact not necessarily be the case. That's a bit of a tangent, but
yeah, it's a great point. It's a great point, and it's very true. We don't know how the animals are perceiving these sounds. We don't know whether they're perceiving all of these sounds, especially when, yeah, the large whales, when they're traveling over tens of kilometers, it's very difficult to know whether an animal 50 kilometers away is responding to a sound that it's hearing.
right, and what the utility of that response is, even if there is one.
Yeah, exactly. Yeah.
Right, another vote of confidence here from Julia Ivanchikova, who says, thank you very much. Great stuff. Such an amazing job. Quite right, too. And the next one is anonymous question. Thank you for your inspiring talk. How do you cope for additional noise coming from other animals like whales or anthropogenic activity? Do you use pre-filtering, a pre-filtering approach to extract only the frequencies of interest? Are you already using this data to extract information about behavior like mating?
Okay, these are all such great questions.
No, questions.
Noise, it can be such a difficult thing to deal with. Some noises are easy to filter out, and yes, we do use pre-filtering before we analyze a lot of these recordings, but other noise overlaps directly with the sounds being produced by the dolphins. And so it can be really, really difficult to filter out that noise. So, there's a lot of work being done right now in adaptive filtering and methods to be able to keep the signal that you want and get rid of the signal that you don't want. But that's all work in progress and not something that I know of that's been developed far enough that we can do this. I can't remember what the second question was.
So the second part of the question was, are you already using this data to extract information about behavior like mating?
Right. Personally, myself, no, I'm not. Most of my recordings have very little behavioral data associated with them. It and that's what you need in order to look at things like mating, but it is possible, and it's something that I'm branching out into. I'm very excited that I'm going to go and look at dolphin behavior around bow riding and look at the acoustics of bow riding and things like that. So there is a lot of work that can be done to look at behaviors and how behaviors are related to sounds that are being produced.
Got it. Short question from an anonymous questioner. Can the dolphins understand different species? I'm not sure if that means different dolphin species or anyway, I'll leave you to interpret.
Yeah, again, we don't know a lot about how the dolphins are perceiving and interpreting the sounds, but if you are wanting to know if they can understand different dolphin species, we do see interspecific interactions happening quite a lot. In the Bahamas, there are populations of spotted dolphins and bottlenose dolphins that occur in the same location, and they interact quite a lot. And a lot of their aggressive interactions particularly involve these burst pulse sounds. And you'll see them facing off against each other and producing these burst pulse sounds. So I think it seems like they can understand those things. And I've done some work looking at mixed species schools. So in the tropical Pacific, we see a lot of mixed species schools, particularly species like spinner dolphins and spotted dolphins. And I've been looking at whether the whistles produced in these mixed species schools are different than the whistles produced by each species when they're on their own. And one of my master's students has been looking at that, and she's found that there are some whistle types that seem to only be produced in these mixed species schools. So this is an exciting area to pursue.
So at the risk of, I don't want you to go out on a limb, but I mean, is there a hypothesis around why that might be?
Yeah, so the behavior in mixed species schools might be different than it is in single species schools. So if they are foraging together, they may need to communicate about foraging, about maybe staying together as their own species or mixing with the other species. So there are things that they need to communicate about when they're with another species that they probably don't need to communicate about when they're alone.
Right. Fascinating. Okay, slightly different question here from Leila Fanfarillo. Thank you so much for your talk. I can't wait to start at St Andrews this fall for biology and psychology. What tools or skills would you recommend for early career researchers interested in bioacoustics? For example, coding languages or book recommendations?
Oh, great question. Well, welcome. I look forward to meeting you, Layla, when you start, and I hope you'll consider applying for my VIP. But for one of the biggest skills to have is coding, like you mentioned. We focus a lot on teaching our students how to use R in many of the biology modules, and R or MATLAB, things like that. If you're not afraid of coding and you're not afraid to learn, that will take you very far in this field. As far as book recommendations, gosh, the list is long. There are some great books out there. Marine Mammals and Noise is the classic book. There's also a new series that is just coming out. There's volumes 1, 2, and 3, learning about animal communication using acoustics. This is a book series edited by Christina Irby, and those are also great books, and they are available. They're open source and free to download, so they're a wonderful resource.
Great, thank you, and thank you for your question, Leila, and I hope preparations for September are going smoothly. I think the second half of this next question you've already addressed, but the first half not. So again, anonymous, do male and female delphinids from the same species produce different types of sounds? Do the sounds change with age? Maybe.
Great question. So you guys are hitting on all the things that we don't know enough about. So as far as males and females go, I think, to my knowledge, males and females produce very similar sounds. And whether the sounds change with age, again, this is a really difficult thing to study because we're not following one population or one individual through their lifetime. And so it's difficult to collect enough data. For example, if you record a calf, that's great. You record it once, but it's in a group with all the other animals. So how do you know which animal produced which sound, which were the calf sounds and which were the adult sounds? But recording animals in captive environments can provide this type of information. And so we can look at things like signature whistles. Signature whistles are individual identifiers that bottlenose dolphins produce and some other species produce. Each individual has their own unique signature whistle that is a learned whistle. And there has been some work done by Leila Sayegh and Vincent Yannick and Peter Tyack and their colleagues on the development of signature whistles through a dolphin's lifetime. And this is a learned signal that does change while they're learning it, but then remains constant through their lifetime.
Thank you. Right, we've got 3 minutes left. I think this is probably the best bunch of questions I've ever seen, one of these. We've got 6 waiting, so I'm going to try and get through them. So you've got 30 seconds on each of these.
Oh boy,
Julie. Okay, go. So DJ Sears says, could you distinguish between a number of the same species all making different sounds versus one example making multiple sounds? Although they say this is similar to a question that was asked as they were writing that.
Yeah, so I think the short answer to that is no. This is something that we do need to, the vocalization rates change with group size, species, location, time of day, behavior. So this is not something that we have good data to be able to say for sure. This is a lot of individuals or a few individuals. Only if you can localize acoustically can you do that, which is possible, but challenging.
Okay. Do seasonal changes in water temperature and density have an effect on the recordings?
Again, no, not really. The characteristics that we're looking at in the sounds aren't impacted as much. The changes aren't great enough to really change the characteristics of the sounds.
Okay. Do the seasonal variations in levels of sound recordings, in other words, in the frequency of sound recordings, reflect migratory activity and or mating or calving events?
I'm not sure I fully understand the question.
So I think it just means that there's when you're seeing more activity or more noise,
yeah.
Does that correlate to migratory activity or reproductive activity?
Sure. Yeah, it definitely can. dolphins will move around. They follow prey around. They like different waters for calving sometimes, and other of their life activities. So for sure, if you are detecting more sound or more dolphins in one area at a certain time of the year, that could definitely be related to particularly the availability of prey.
Are the dolphin populations around the Scottish coast in decline, or are they fairly stable?
So we haven't been able to look at all of the species in detail yet. This is what our new classifiers are going to let us do. The bald-nosed dolphins seem to be doing quite well, although it looks like their distribution is shifting slightly. They're ranging further south than they used to. So we don't know yet whether that's a change in population size or a change in migration patterns, or what that is. But yeah, stay tuned for more information about that.
Okay. We're nearly there. You're doing great.
The pressure.
Trace Lasky, is there a wider classifier network beyond the locations you mentioned? And have these classifiers been applied to other marine noise, such as reef fish? Thank you for the talk.
Great. Well, you're welcome. There are lots of classifiers out there. Classification work is something that a lot of people are doing. Many of the classifiers, though, are not available easily. And so if you search hard enough for the area you're in, there might be a classifier available, but more often than not, there aren't. One of the things that Tristan did was to do a review of the literature and look at all of the classifiers that are in existence, and that publication will be coming out very soon. So you'll be able to read about that in greater detail. But you know, there's a big cluster of classifiers for the tropical Pacific and areas around the United States, and then areas in the Southern Hemisphere are much leaner on the classification side. So I hope to plug all of those holes.
Thank you. Right. Last question. We're over time, but I'm determined to get in because this is another very good question. Great talk, Julie. Thank you. Oh, where's that gone? Sorry. Referring to the figure where you showed the peaks in vocalizations in the dolphin species, was this 6 p.m. and 7 a.m. peak consistent over a long period, or did it vary? I.e., was there any connection to tides, for example, for increased feeding activity?
Yeah, so the data I showed was for all of the locations and for all of the times of the year. We don't yet have a large enough data set to be able to break it down like that, but I hope that by applying the classifier to more years of the data, there's quite a large data set available, then we'll be able to dig into things like that, and that would be a really interesting thing to explore. Fantastic.
Thank you. We've done it. Thank you so much for all your questions. That was really terrific to see so many insightful and incisive questions there. And thank you, Julie, for your responses and for getting us through them, and for your fascinating talk. We will reconvene for the next Saints talk on the Tuesday, the 24th of June. Which will be given by Dr. Matea Peter from our School of International Relations. Matea is Executive Director of the Scottish Council on Global Affairs, which is the only UK foreign affairs think tank based outside London. And her talk is going to be titled Transactional Peacemaking, What to Make of US, Russian and Chinese Approaches to Conflict, which feels pretty topical. So we hope to see you on the 24th of June for that. But in the meantime, very many thanks again to Dr. Julie Oswald for her talk today, and to you all for joining us, and a very good evening from St Andrews.